field of message is even wider. So we have to teach our students not only this is the molecule of interest, but we also have to teach the physics, the chemistry of the detection methods that we have in order that we can really measure what's ongoing in the cell. So we ask our students really practical competencies, but we also ask to understand the biological background, and we ask to understand kind of everything which is in natural sciences, and even more sometimes about the machines that do that in the end. As this is also a trend, especially in this field, that we really have to think about computer capacity, stochastics on all these data. So handling big data sets will also increase the need for students that have a good understanding of statistics and stochastic in the bigger field. So let's give some illustration on the method types that we do have here. So first thing is the PCR. So the basic principle of PCR is an amplification using specific primers. So we always start with a template DNA, having those primers here, and there's a number of cycles where you do, so I make it very short, where you do an amplification. You do 20 to 40 reaction cycles that starting from, in theory, one starting molecule of DNA, we end up with a very huge number, as this is exponential, of copies of that molecule. And that enables us to really see whether a gene is there. We can use that amplified DNA in order to transform it into vectors for genetic manipulation of cells. We can do a big number on that, and we can even work on RNA. The only thing that we have to do is to translate the RNA back to DNA, and there's enzymes that are capable of that. Reverse transcriptase, so that you can also do an amplification of RNA in a cellular context. In the very early times, this was done kind of as a endpoint process, but today we can use two approaches. The one thing is to apply everything on an agar scale to just see, okay, the product is here or it isn't here. But we also have the possibility to do quantitative analysis, which we call which we call qPCR, quantitative. That means that we use fluorescence to watch those cycles and the rise of the amplicon, the amplificate, to be seen in here, and by that we can derive the number of copies in here. So that is also a very strong development during the last years, that we started with qualitative description of the genetic background. Most of us who studied biology in the 1980s and 90s, we still learned how to make DNA maps and so on, and that was sequencing base by base, and everybody was talking about the Human Genome Project, which basically took 15 years to deliver the genome of a human. Today, you can get your own genome, let's say in a few days, and that is really an improvement in the possibilities that we have as biologists in order to describe systems. So this is the basic principle of the PCR, and you can use that amplified DNA for measuring the amount of a specific genes, but you can also use that for sequencing the molecules in here. So that means there's kind of a very big number of possible outcomes of these amplifications in here. And today we do talk about sequenced genomes. Of course, we talk about bacteria and viral vectors. But, and this is actually the next trend, I would say, that we face in the molecular field, that we have very good options to synthesize DNA. So I would also predict that in 2 to 3 years, nobody will go and isolate small amplificates. So everybody will just buy the DNA done by synthesis. But however, we go for PCR, and then we can use that in order to design probes to find the genes in their context. So I will come back to that when we talk about the hybridization methods. So, well, first you do an amplification, and then later you try to see that. The second one I already indicated is the quantitative use of PCR, which can be applied to show contaminations in food, but can also be used to show COVID, which we probably will hear tomorrow. But of course, it is still being very valid to do agar scales in order to study the structure of DNA and RNA. So the big trend in the last years was finding silencing RNA, so regulatory RNA. So, and that was really kind of a push into the direction to understand functionality. And of course, we have all the kind of sequencing technology in multiplex and single gene in order to use that for diagnostics, but also for function thing. And we have as a big branch still all the molecular techniques that we can use for changing systems. So when we talk about production organisms, starting from Escherichia coli being transformed to produce insulin, but ending with gene therapy, I would say that is kind of the next big issue we also have to discuss from a responsibility point of view. So as a biologist, I can just say, I describe the system, I give you the tools, but from the point of a responsible scientist and of a citizen of this world, I also have to discuss with my students where are the moralic, where are the ethical borders of what we do. So are we allowed to change the genetic background of the systems? Aren't we? And I think this is kind of, without giving a prejudice in there to say, okay, you are not allowed to do that, or you are allowed to do that. But I think we also have to train our next generation in order to be able to think about the consequences of the changes that we do here. So again, challenges and probably obstacles that we have in that process. So teaching molecular biology is not only techniques, it's not only functionality of molecules, it's also being responsible scientists with respect to genetic modification and changing probably evolutionary processes that we face in here. So this slide has some issues, but I want to change from PCR-based methods to the hybridization techniques that we do have here. So the basis is, again, that you do start from a sample that might be a sample of your bioreactor with some microorganisms in here, but that might be also something that you derive from cell culture, that you isolate from any kind of organism in here. And of course, you have to extract the molecules of interest. When they start on RNA, then of course you can think about transforming it into DNA again, or we do start from DNA, do a polymerase chain reaction in order to have enough material that you can see in here. So, and then we have hybridization methods that we call dot plot hybridization, where we do have quantitative hybridization with labeled probes. I will come to that in a second. You can minimize the amount of it. This is what we then would call an array with many small dots actually, and that might be done on membranes, but that might be also done on glass slides, and that can be automated. So microarray was a very huge trend in the late 90s, but with the new, let's say, new sequencing technologies, these blotting techniques become less important on a level of DNA, but they are still very important on the level of RNA. The other option is to do something with the whole cell, so doing whole cell hybridization in here those are techniques that can be used to visualize the spatial distribution of molecules in a cell. The basis of all these techniques is complementary base pairing in DNA and RNA level. However, for that, you have to label your molecules, and you do see that we have two big options in here to do labeling. So, and in the early years, that was mainly done with radioactive phosphate, and of course, again, responsible scientists and even scientists who want to stay healthy. We, of course, try to avoid everything which is connected with radioactive labeling. And we rather use kind of colors. We use molecules that can be detected by, for instance, antibodies, or molecules that can be detected by very specific binding. For instance, avidine, the avidine example, where you have a very, very strong binding of two natural molecules that if you attach them to a DNA or RNA molecule, you can use it as an anchor that can be used for detection. So we can either start from the purified DNA fragment, in most cases that's a restriction fragment, and then you do kind of some amplification in here, and the DNA polymerase in the end polymerase chain reaction will incorporate labeled nucleotides. So you would do the labeling on the level of the nucleotide being incorporated into your molecule of interest, resulting in a population of DNA molecules that contain a labeled example of the sequences on both strands. As of course you run the PCR, the whole cycles, and you end up with labeled things. The other option is to do an end labeling, and the nice thing is that you can really use, and this is the example with the radioactive phosphate here, but of course you can use that addition via a phosphate bound with a fluorescent marker as well, and you will kind of use the polynucleotide kinase again, an enzyme system available, and of course a biotech product, in order to do an end labeling in here. So that means in the very end, you do have your desired DNA fragment with a single strand labeled on one end. So, and we imagine now not having the phosphate here at the rear end, but having any kind of colorful fluorescent dye attached to that. So that means on the other hand, when we talk about molecular techniques, we also talk about the advances in, for instance, organic chemistry. So having many different fluorescent markers available today enables molecular scientists to do multiplexing, to see more than one gene at one approach. So that was the big step from in Sanger sequencing, even when we came from the radioactive labeling into the fluorescent labeling. And this is the big step forward for all kind of visualization techniques in both sequencing, but also when we talk about microscopy in that field in here. And this is the next example that we do have in here, where we apply fluorescence in situ hybridization. What we do is we take our cell, and within our cell, of course, we have the ribosome, and we can work with the RNA of the target organism. In that example, we focus on taxonomy of organisms. That's why we take ribosomal RNA, but you can imagine the same procedure in order to show other molecules. Ribosomal RNA is very interesting because you have high numbers of copies in there, as a living cell need to have a number of ribosomes, and with that, of course, a big number of copies in here. We use a probe which is labeled fluorescent with fluorescence, so that's why fluorescence in situ hybridization, and we make that probe entering the cell. Of course, having an RNA molecule to be going into one cell means that you really have, in the end, to kill the cell without destroying the cell, because we want to see in the end, that's a microscopy technique. In the end, we want to really see where the cell is. So what you can see later on is something like that. So you will use the staining of DNA in general, and then you use your probe here, which is specific for an RNA fragment, in that case of E. coli in that figure, and you do see all bacteria that are in the sample. As you see DNA, wherever it's DNA, you get a bluish signal, and you get that red signal wherever an E. coli is in your sample. And then you can do an overlay in here, and you do see, okay, there's E. coli in the sample. So this is what I mean with respect to microscopy, having in here. So, and then you can, of course, do multiplexing with different kind of probes. You do see every different color here is referring to another gene. In that case, a surface molecule that is on a bacterial cell. So that's why the quality is not too big. So we had to really enlarge that figure so that you can see different surface molecules by means of different fluorescent markers in here. The basis is hybridization in here, but you can also do the same thing when you use antibodies coupled with a fluorescent marker. The principle is the same. So we can also transfer the basic idea, the basic principle in here. And thinking about teaching, I think that's also important to think about, where do we have very easy to understand basic principles, and how can we teach them very deep, and then later the student is able to see, I've seen that before. I saw that with DNA. Now I do see that with RNA. But as I understood the basic principle, I can do what we call a transfer of knowledge, and then I can really dive into that with my basic knowledge. And I think these are very important features for the students, as I illustrated that the speed, the velocity of change in methods, especially in the molecular field, is so fast that teaching today is already old teaching, because of when our students enter to the market and get their jobs, probably the methods that we taught them are no longer valid, and they have to we have to prepare them for a changing method way. However, as long as we teach the basic principles, they will be able to adapt to that new challenges that they do have in their future. And I really want to invite you to get that thinking on what might be relevant in future, and what is the basic principle that you do. This slide is just to illustrate that, of course, you can do a number of combinations, and that number of combinations will, in the end, give you a more throughout picture of the functionality of the molecules in our cells. But we just skip that. You can use a number of examples, for instance, for the functional description of microbiomes. So that is very common now in order to use as an example, to focus on gut microbiome, for instance, and then to explain, okay, you can go the path of DNA, you can also do the path of RNA, but in the end, you have to bring everything together. And I would even go one step further. I would always say, okay, it's nice to see DNA and RNA level, but you also need to see the proteome level, and even more, you do need to see the metabolome level, and then you will get kind of an idea of what's going on. Well, I always warn my students, so if you have a look to those figures, you have the feeling, we know everything about the cell, and we can just turn one screw, and then we can predict what is happening with the cell. We still face the situation that biological systems are very, very complex, and we are far from understanding really all interactions, all possible interactions, especially when we have the cell in a changing environment. And that's sometimes the frustration of the students. So they learn the theory, then they go to laboratory and they learn, sometimes the biological system is behaving, let's say, minimum interesting, but not as predicted. And then we have to think about, okay, is it a problem of the method, or is it something that we don't understand? Because biology is complex, and I would also add wonderful and still surprising. And this is actually what is triggering me as a biologist to continuously being interested in that amazing field of possibilities. So, yeah, I just wanted to illustrate by that example why so many people have to talk about molecular methods. So one thing is, of course, that we can really understand cells on a systems biology level. And the second thing, of course, and when it comes to diagnostics and application of the methods in the end, this is the thing we have to look at. Yeah, so when we do culture-based identification of microorganisms, for instance, yeah, so that's the optimistic 3 to 7 days time frame that we have, sometimes a bit faster when we talk about E. coli, for instance. However, the time to result is quite long. And when we just switch into molecular techniques with that example for the identification of fungal contaminations, then we either stick to 2 to 3 days when we go classical Sanger sequencing. But however, when we do a specific PCR in here, being able to quantitatively deliver results, we end up with 1 day. And of course, having the pandemic in our backs now, thinking about how important it is to see whether we have infected or non-infected persons, then of course we know that time is crucial, not only with respect to money, but with respect to health and even survival of persons in here. So with that, I want to close the initial part and do some more striving on the teaching in here, how to do that specifically. So with that, I want to invite you to go one step back and think about the teaching. And we really have to think on 2 levels. One level is the teacher-oriented aims. So this is that part here. So why should we teach molecular methods? So what should the students know? What are the competencies in there? What's important? However, we also have to think about, and I think that's very beneficial when we think about the students, to think about the student-oriented aim. So why should they learn that? So what is the benefit of knowing it? What will they need for their job? So having the sense of it and how to apply that. So I think there's different methods in teaching in that, as of course, if you go the teacher-oriented way, then of course it's always about, this is the knowledge and everything is important, but vice versa, the student will be limited in taking up things. And that's why we have to really focus in here. So that means we have to align teaching and learning methods with respect to the intended learning outcomes. So, and I think it's obvious that you have to use a mix of methods in here. And I want to give you a short homework for when you start thinking about your own teaching. So that's always to start, okay, from your own point of view, try to illustrate what are the teacher-oriented aims and what are the students-oriented aims and what are the needs in the fields of molecular methods. And as I already indicated, when I reflect my own teaching in here, I would always say the need is that you understand the basic chemistry of your target molecules. And the second need is that you need to understand that the methods are changing so rapidly that the most important thing is that you get the basic principles of those methods. Because if you understand them in very deep detail on how to change them, you will be stuck in the old methods. Being an expert, perfect, but not being able to continuously develop. So that is another need, and that in the end, that it's a need for creativity, and it's a need for a mindset which has nothing to do with molecular biology. And this is sometimes a very hard thing to see as a teacher of molecular biology, that it's not about molecular biology, but it's about how to be flexible, plastable in your minds. So I want to show that to you, how we do that in our teaching programs. And as I already said, we have two main teaching programs in here. A bachelor program where we do an introduction into natural sciences in the first 2 semesters. And I think this is the basis to teach the students the idea of the chemistry of the molecules. So they really need to understand organic chemistry. They need to understand instrumental analysis principles, kind of what is a photometer, what is absorption, what is fluorescence. They need to understand on a quite abstract level, and we will come to that. Abstract learning is something that is very late in understanding. So we always have to think about, okay, how much example we do provide versus how much abstract knowledge we do see. And many young students learn example-wise. Major students rather go for the abstract level, but that depends from person to person. Some of us always want to have the example, some of us go to the abstract level. Anyway, our students have to decide between bio and food technology and process engineering, and of course the deep detail is in that specific teaching. And I'm very happy that tomorrow we will have an example as Holger is going to show us how he's teaching on that level in here. The specific thing with our students is that they do their bachelor's thesis directly in industry. So we have to prepare them before the 7th semester that they are as major as to go into any company and see, okay, what are they doing? What are the methods? And how can I bring that together with what I learned before that? That's even more demanding when it comes to the study program in the master's field, where we have a 3-semester Master of Applied Bio and Food Sciences, where we have a first semester on theory, and then we directly go into problem-based learning approaches. I will come to that. I think that is the big issue for master education. No matter whether we talk about molecular techniques or something else, we have to bring our students into the situation that they can really dive into a variety of different topics, but learning the basic methods and how to find out what's important in a new topic, how to get new knowledge, and I would even say how to do lifelong learning, so how to frame that on the student level that you are able to learn during your whole lifestyle. And I cannot stress it often enough. We have a very huge responsibility as teachers because we are the role models. And when we are able to show that we do learn our whole life, and that is good to do so, and that we are not kind of the experts and everything is sad, but that learning is something pleasureful, we can be a good role model for our students in the future. So starting with that, you should take your curriculum and think about, okay, what are the application fields? What are the topics in here? That's the example of our master study program. And what are the skills? Because of this will guide your teaching. And of course, there's a number of things that I can teach in molecular techniques to my students. I can do everything on DNA structure, on structural biology. It is super interesting. But with respect to the application field of my students, that's not needed. So they rather need to understand how methods are working, because of they will work in product development, in quality management, and in process design. So always think about, okay, what will they need? So that's why I suggest to do an approach for your specific teaching. And I do show that with the example of molecular techniques. So it's always kind of think about those headlines. In your field, what are the big ideas? In your field, what are the experimental approaches? In your field, what are the current controversies? To really show that science is nothing fixed, it's not ready, so it's developing. Yeah, and always, and this is quite interesting for students because of this will trigger their curiosity. What are the non-solved questions of the field? I do give you the examples. What I do think in molecular methods are the things that I put up front my teaching in order to see, okay, what do I have to reflect in my slides in the end, being in the classroom. And I would always say, okay, in molecular techniques, the big ideas today are that we talk about holistic systems. So we cannot just focus on DNA. We also have to focus on RNA, or we have to include the idea what is happening on enzyme level. And the second big idea to be taught is that interaction of molecules is the basis for all kind of functionality on the organism level at the end. When I think about the experimental approaches, I also think about, okay, what do we need to taught the students in the laboratory work? So, and I would always say, okay, it's how to handle the biological molecules, that they are not destroyed during handling. I think we have to talk about sequencing as one basic technology. And it's not only PCR, which is the basis for that, but sequencing and handling sequencing. And the third thing is that they should be very comfortable with instrumental analytics in there. And the current controversy is that if you go to literature, you will find out people that say, yeah, I did the DNA story and I can explain everything. And then you go to an RNA story and they tell you, okay, we can explain everything. And if you compare the results, you will find out that they have some overlap, but there's some, let's say, truths that you can derive just from the DNA level, and some truths that you can just derive from the RNA level. And we do not see the holistic picture in here, but still people, of course, have their preferences in doing so. And I think there's a lot of controversies in that field. And there's also a big number of non-solved questions for future. And I would always suggest the students to develop into that directions. And I would say one non-solved question is how to handle systems complexity. So probably that is a non-solved question forever in biology. That might be the case. And the second thing is how to handle these huge amount of data on a single data point level. So if you do sequencing with 300 times seeing the same base, you cannot evaluate your data on a single data point thing. You have to think about stochastics in here. And of course, you need to be quite good to handle that data. And I think we are far away from doing that in a proper way, how to exchange data and so on. So that is future.
And sorry, about 5 minutes more.
Yeah. So and then I come to my end. So the thing that I want to show you at that point is that I think it's good to think about your curriculum with respect to what should be knowledge and what should be competencies in here. We will always say that you should have kind of a delimiter of 1/3 being knowledge. So, for instance, learning species names, morphology descriptors, or in molecular biology, what is DNA, what is the structure of the molecules. And 2/3 is the competency. So how to classify species, how to do that. And this is what I wanted to stress before, that from a teacher's perspective, you would probably invert that and say, okay, this is the knowledge. So we have so much knowledge and we are always pressed to really focus to a small portion of that knowledge in here. Well, we have to turn that. And that makes you as a teacher rather being a person who is attending the teaching, is learning on the same level as the students. And you shouldn't be shy to say, I don't know it. Let's find out it together. This is the way how to do that in there. And I will end with that citation in here. There's much more to be said. I think this is maybe a motto that you can put on the top of your desktops. So, given a fish, one can eat for one day, but if you are taught to fish, you can eat for your lifetime. And I think that is a good point for thinking about how to teach in every field. So I will end with that. I'm happy to provide also my slides afterwards, which have some more details on problem-based learning and how to design competency-oriented teaching. But I think the more detail we will see tomorrow when we focus to how to do that in lab and how to do that in classroom. And we will also see the example from University of Jember. Thank you very much for listening.
Okay, thank you so much, Anja, for a very informative and elaborative about the methods in molecular biology. And what I find also really important is that you mentioned that we as a teacher is kind of a role model for our students, that how can we deliver to them that learning is something pleasurable, not something horrible maybe. Yeah. This is not easy for molecular biology because sometimes students facing molecular biology something to be, oh, it's just like this, and this is really a very important task for all of us. All right, now we are before the discussion. We will have a panel discussion later on after the second speakers give also the presentation. Now I would like to invite Professor David. Prof. David.
Yes, I am here.
Okay, yeah. Yes, so Prof. David, MSc PhD, he is currently a lecturer at the Faculty of Teacher Training and Education from the University of Jember. He is currently also a dean, so today we have two deans here at our webinar at that faculty as well. And Prof. David has been so much engaged with the didactic methods, so I'm sure we will hear many more about how to teach the student, especially related to problem-based learning and also in relation with our current situation. So, Prof. David, now I wanted not to prolong my introduction. Now the time is yours.
Thank you, Ibu Kartika Senjarini. Good afternoon, everyone. Good afternoon, Angela Bes and the Honorable Rector of the University of Jember, Bapak Iwan Taruna, and all the lovely participants of the online seminar today. Please allow me to share my presentation. This is the title of the presentation on the student creative, innovative thinking skill in solving two-dimensional arithmetic sequence problem through research-based learning. We have three elements of our point of view in this discussion. I'm talking about creative, innovative thinking skill. I will talk about the two-dimensional arithmetic sequence problem, which is a non-procedural problem. And also, I'm also talking about the didactic methodology, the teaching method, namely research-based learning. Actually, my background is mathematics. I'm very much working on combinatoric and graph theory, but currently I am a dean in the Faculty of Education, so also one of my research of interest in education, especially in research-based learning. We have a lot of relevant references that we already published in the international reputable journal. The first one related to two-dimensional arithmetic sequence. We have publications in 2017 until now. And the application of the two-dimensional arithmetic sequence, we have also publication, and we can implement and apply our result in the crypto system technique, and how to do a research and integrate the result into a teaching. And then also we develop a new teaching methodology that we call research-based learning, that we also have this publication, especially in the International Journal of Instruction. Okay, back to our three elements of our talks. We have three elements. The first one is the student creative, innovative thinking skill. This skill actually is very required in the 21st century era. In the 21st century era, people can survive in this daily life. They should have at least 4 C's. What do we mean by 4 C's? The first C is creative, innovative. The second C is critical thinking skill. The third C is communication, and the fourth C is collaboration. So people who have this kind of C skill, they can survive. They can be considered, be able to survive in this daily life. So what is the keyword in the creative innovative is the novelty, of course. It is what is the indicator in the critical thinking. People can ask a question and also can give a reasoning and do analyzing. And in the communication, of course, they use Internet of Things. They use the hypermedia in the communication, not only face-to-face basis, but we need to have online basis. But when we use online basis, we need to be a content creator, use a application software, use a specific platform of application. In such a way, people can interact each other, and as well as collaboration, mean cross-country diversity collaboration, not only within university. And right now, I'm very happy to have Professor Angela Bes and others collaborators from Germany that can be develop a joint research in the future. So what is actually the level of creative innovative compare in a Bloom digital taxonomy? So actually, when we talk about education, normally the level of cognitive can be measured by Bloom taxonomy. This is a level of cognitive when people and as well as teacher implement and interact with their student, and the learning outcome only applying, even understanding, and only memorizing something. That's considered to be lower order thinking skill. But when we ask student more, even more doing something in the laboratory and ask the real thing, demonstrate the real thing, and as well as develop a new thing through a research, for example, join research within a research group member, then this activity considered to be HOTS, higher order thinking skill. Creative innovative in Bloom taxonomy is considered the highest level of cognitive that can be pursued in such a way the student will be productive and also will be productive. Now there is the revised version of the Bloom in digital perspective. This is a Bloom digital taxonomy. So when we only doing download and download all the time, we are only a customer. We are not a producer. If we only doing a copying and copy, so only the lowest level that we can do. So, but if we do a researching, for example, surveying, grading, testing, and filming, postcutting, and directing, that's actually the 21st century skills are required in nowadays life. So when we are teaching and when we are researching with our student, so bring them to the highest level of cognitive. That's logic. It's not enough. It is yes, and after that, critical and creative. Even the highest level beyond of the Bloom taxonomy is metacognition, metacognitive. That's we now deal with this kind of learning objective or learning outcome when we are doing a research and integrating research into a teaching. So what is the creative, innovative, which is part of the 21st century skill indicators? Creative, innovative, of course. Student can demonstrate originality and inventiveness. That's I call novelty. And the second one, student can do questioning, analyzing, reasoning, arguments, interpreting, and reflecting effectively. That's part of the indicators of critical thinking skill. And also communication skill, articulates through and ideas using oral, written, non-verbal skill in variety of form of context, and also collaboration skill, of course. We need in this today era, because without collaboration, how strong people, if they just do and carry something by themselves, I believe they cannot be. So the keyword, the main indicator, a part of 4 indicators of the creative innovative skill is a productivity. Productivity is measured by the number of production, also the number of achievement, and as well as speed. That's the main thing. Even we can do something, we can publish something, we can write a book, we can write a monograph, but we just write a monograph 1-2 years, for example. It is not productive people. So in the skill, people also can be assessed by productivity, even they are creative, even they are critical people, but they are not a productive. It is also do not consider is possessed and creative innovation. Now, with this kind of learning objective, we try to implement in our classroom for every research of interest. So in this kind of topic, we try to do and run as class and subject courses, doing further into a detail, so teaching something deeply, not widely. This is the main thing. Teaching something, teaching subject course deeply until they can find the state of the art. of the knowledge, the state of the art of the science. I also ask to my colleagues, don't teach everything to them, because everything can be learned individually by a student, but teach them until they can find the state of the art, until they can do a research about that topic. So, for example, I have here, when I try to talk about, to teach about something very, very simple. I'm a mathematician. Start from the simple one, is arithmetic sequence. I believe this arithmetic sequence already taught in elementary school, in secondary school until high school. Even in the lecture, also repeat this subject course in the higher education. It is very simple. But how do we bring this topic into a deep understanding to a student until the student can find the application in the real life and can do a research of this one? This that we call a research-based learning. So student do a research in a classroom, and teacher and lecturer drive a research in the classroom for the subject courses. The question is, it is possible for the lecturer who drive subject courses who has laboratorium activities. For example, in mathematics, some of the lecturer having a research in a grounded theorem. So they develop a theorem, they develop a technique, they develop it. But we also develop an application of this one. And now by research-based learning, we try to give a student the simple one. Okay, student find out the arithmetic sequence by filling the tables. It is start from the empty tables. So just consider the red colors when we say 1, 2, 3 in rows, and follows 4, 5, 6 in rows. And student can generalize. It is the easiest one. Okay, now apart from starting from the left side into the right side, and now we are starting from top to the bottom level. Top level 1, 2, 1, 2, 3, 5, 6, 1, 2, 3, 4, 5, 6, 7, 8. Okay, that's easy. In the classroom, maybe even we use online base, we just give this one to a student material through the presentation or slide presentation in this pandemic. It is easy to understand. But how if we try to generalize this one? Generalize mean can find another structure of the this kind of arithmetic sequence, or we can find another permutation of this one. So the student is challenged to find other structure or other permutation. So this is actually, we give them a proposed problem, we propose them a problem with a complex problem which cannot be solved by programming. Then we consider this is NP complex problem. It is not, there is not exist the polynomial time for the solution. Now, students are encouraged to find another patterns, another structure which is different between student to other student. If we have 35 students in the classroom, and then we ask them, find out which is different with your friend. So there are a lot of combination. Trying from this combination, finally, we generalize them. This is the most highest level of the creative, innovative, because when we generalize from the certain case, maybe we couldn't find out the solution because only one case. But if 35 students having a different solution, and then we can generalize based on their job, based on their result. So in the classroom, we have an activity of research, not giving them, not introducing them a concept, a procedural, a technique, but students find out a research, find out a technique which is different with each other. And the last, how to bring this one more than that, a knowledge in the classroom. Research group members of the research, specific research group in this program study or in the university try to find out what is the application of this one. So actually the application of this one in our subject research, we call anti-magic labeling. So actually it is related to Angela's. Last time we tried to find out the magic labeling of DNA. Yeah, so this is, now this is the deeper. Actually, it is not actually in the subject or topic in the subject courses normally. But we bring this kind of topic a little bit further until they can touch the state of the art of knowledge. Not bring them a lot of things about information about the arithmetical sequence. So when we have this kind of labeling, then we can find any application of that. For this case, we find the application of the labeling based on the arithmetic, two-dimensional arithmetic sequence into crypto system. That we have a process, two process, encryption or decryption. We already published this in the good reputable journal. How this is work? For example, in the labeling and the crypto system which is convert plain text to ciphertext or ciphertext to plain text. So we have two things, encryption and we have decryption. So we give them to our students, which is beyond of actually in our topic. So, but at the beginning, our team developed the learning outcome of this kind of subject courses and bring these subject courses a little bit further, and such way the student can do a research. Now, so for example, we have a plain text. The plain text is going down FKIP University of Jember. By this kind of labeling that we have generated from the two-dimensional arithmetic sequence, and finally we have ciphertext, GMRBICHR. Actually, the plain text is FKIP on it, but because of the encryption process, we have this kind. This considered to be complex crypto system, which cannot be revealed by unauthorized people. So because of this one. And the student now about the fundamental concept, and the student now about the application, because a highest level of the Bloom cognitive taxonomy, namely creative innovation. But if we just only ask students to do applying this one, calculating this one, determining this one, I cannot guarantee that the student can do a research best for the best productivity, because we do not bring them into a highest level of Bloom taxonomy. Now, so this is the thing that I can guarantee when we try to have about combinatorial problems, I can confirm which indicator of combinatorial problem can meet the 21st century skill. Look at it. So actually in mathematics, or guarantee that all the problem, the first problem in combinatorial problem, which is related to the arithmetic, two-dimensional arithmetic sequence, does the solution exist? Student fill the table, which is starting from empty table. Can student fill the table in such a way the addition, the summation of the column form an arithmetic sequence? That's the first question. And if we perform this one with the 21st century skill, that's, if student can do that, that's innovative thinking skill. So all those kind of problem actually associated with the 21st century skill. Now, I try to have break down the detail of the creative innovative thinking skill in two-dimensional arithmetic. This is the side of the education. Creative innovative thinking skills, student can find other pattern. And also creative innovative thinking skills, student can find a new pattern, can do a local generalization. Everything, I break down this one in such a way we can characterize individual student and other individual student for their portrait face of their creative innovative thinking skill. This is in terms of teaching practice in the classroom. But it's okay, but I don't want to go further into this one. The last one, what is the research-based learning? Research-based learning actually adapted by IDB project last time and already implemented in this university curriculum that we call research-based curriculum. So actually research-based learning in terms of the curriculum element, it is a process standard. The curriculum in whole is a research-based curriculum. You can find in the Google, it is terminology already there, and somebody already published this kind of topic. So what is research-based learning that we should perform, and we should know this one? I believe in the university and abroad, especially in Germany, UK, Australia, already aware about this one. So there is triple helix relation. First of all, faculty members, of course. Second one, student in the class. And the third one is the most important thing, is research group and research society. So this main thing is very important to deal with the productivity, either in publication, in book writing, in scientific research, and as well as in the classroom. So the main thing of the research-based learning, which is considering the three elements, is bring the result into a classroom. I always ask to my colleges, don't bring other paper publication. Bring your paper, your publication into a classroom. Even you should bring your book written by you into classroom. And your student will be proud of you. Otherwise, if you bring other paper result, and your student then proud of you. So this is important, faculty member. Now, the second one, in the research area, in the research perspective, when we implement the research-based learning, we should aware a student research is a lecturer research. Sorry, Aji, might be in the university in abroad, it is already aware by some other people. But in Indonesia, it just started right now that Research student is research student, lecturer research is lecturer research. But now we try to combine because we should have a joint research. Even we are a big professor, we are a full professor. Without any student, we couldn't do anything. But I just started to give this kind of a strong recommendation to my researcher, to my colleges. Your student research is also your research. And you have a joint paper, you join co-authorship. It is very useful. Otherwise, you can do it productive. So, 2019, University of Jember already established a new rule of the rector and perform a research group from starting from the department until research department level, faculty level, and even university level. So Anji can see when Anji in our university, there are a lot of, there are SIDAs and every other that Ibu. So my research group member also called combinatory graph theory and network topology. It is TA, graph theory and application worldwide. And Indonesia, we have also a society. And in department one level, we have this one, but I don't want to have this one. But okay, what is research-based learning? Associated with the existence of the research group, this kind of process should exist in the classroom. Learning process actively involves students to do a research in solving a problem arising from the RG open problem. See, underlining that's our strategy now. Last time, when a lecturer give a problem to a student in our faculty, they generate the problem from the internet. They generate the problem from the textbook. They generate a problem and give it the procedural problem. And now, no more exists. The lecturer give a problem from the textbook. The problem should from the RG open problem. What the RG member already done, already published, of course, in the last, after conclusion, they give a concluding remark that in this kind of paper, there is something open problem that which is not have done yet, and this will be good for further researcher. They bring the open problem into a classroom. From this one, the classroom start something, a partial solution. When the classroom start to find out the partial solution, and the lecturer, which is a member of research group, bring that partial solution into a research group to have a focus group discussion. The latest one, the result of this one, they bring again into a classroom, so there is a mutual symbiosis between classroom, lecturer, and the research group member. So that's why we develop a new syntax in the research-based learning, which is already published in the Journal of Instruction. This is the research-based syntax. Problem posing arising from research group, open problem. Students are encouraged to develop a problem-solving strategy. Just normally like the procedure of the having a research, orientation data, they collect data from internet, because from social humaniora point of view, there are a lot of data in internet, data analysis, and they do a RBL report. This is in the classroom. How to bring the RBL report into a research group activity? We choose which a good RBL report from the classroom. We choose one of them, or two of them, three of them, which is potentially can be followed, can be extended into a real research activity in the laboratory. So the beginning is the problem, the partial solution rise from the classroom. Finally, the research member selecting which RBL can be potentially to be a paper of research, this one in blah, blah. And finally, we have a publication only from this kind of thing. So there are two things that we can have a benefit of implementing the research-based learning into a classroom. The first one, we can give a deep understanding to a student in such way the student can reach the state of the art of a certain topic. Not wide, but deep. That's the key thing. And the secondly, we can utilize the result, the report from the classroom of the student and bring to the research group and finalize and analyze and extend, bring to the lab with the research group member, of course, with master degree program, postgraduate degree program, and then we can have a publication. So with this kind of thing, sorry to introduce this one, we have this achievement, a SIGAN research group from 2010. We only have less than 20 publications, and now we have more than 160, and altogether maybe 400 publications so far because of implementation of in the class. And I believe this one is not. Now, because of pandemic, and government also encourage and recommend to study from home, now the interaction with the research group member, we mostly using online basis for doing FGD, by using Zoom cloud meeting, and also Big Blue Button, Microsoft Teams, and Google Meet and everything. And also we have a content creator, research topic identification first, and hypermedia-based research worksheet. And finally, research process, even we do not make because in social humaniora, especially in our faculty, and as well as in mathematics side, we do not have, we do not need a lot laboratory. It might be different with the Angela Bes and Ibu Kartika. So we can easily doing FGD workshop, colloquium by using online. So that's why this one, that strategy in the pandemic situation, we develop a learning material by using simple tutorial as a screen recorder and developing slide PowerPoint presentation by inserting audio and video features, and also developing a slide presentation by using Canva, Camtasia, Screen-O-Matic to give them, to deliver material. Even we use software by podcasting by using green screen and using application Filmora. And even we also insert QR code with audio with page. Then the last one, this that we also use right now, a Zoom cloud meeting using a brick house system for hosting and everything. And our leader rector also recommend to use this kind of thing. Thank you very much, Ibu Kartika. Hopefully this presentation is benefiting to all of you, and I'm looking forward to having a discussion. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Okay, Waalaikumsalam warahmatullahi wabarakatuh, Prof. David. Thank you very much for a very nice presentation. This is really challenging for us, especially for researchers, to bring our research product to the class. Yeah, sometimes we are still feeling under, how can I say, underestimate if we see about our research results compared to the other scientists from the other country, that so that it is not that easy maybe for us to bring our own result, all of it to the class. But you give us really a good example about it. And now I'm very eager to have a discussion. Some of the participants are already giving a question in the Q&A, and I think Anja has been answered all the question. Yes. So I would like to invite the other participants who haven't wrote in the Q&A, just raise your hand. Okay. The co-host, please help me in giving Ali Wafa, Ali Wafa Faperta, giving them the to promote them as a panelist so that they can talk. Or maybe I can. Yes. So Pak Ali Wafa, you are now allowed to talk. Pak Ali Wafa. I'm sorry, Prof. David, can you turn off the slide share so that we can see directly the...
Yes, yes, sorry about that.
Okay. Pak Ali Wafa, are you there? Yes. Pak Ali Wafa, I'm already promote you to a panelist, so now you can directly ask the question. Yeah,
okay.
Pak Ali Wafa. Yeah.
Thank you, Ms. Kartika.
Can you please turn on also the video if it is possible?
Okay.
Yeah. Okay.
Yeah. Thank you, Ms. Kartika.
Yes.
I have two questions for Prof. Anja. I hope Prof. Anja still remember me. And the question is about the basic molecular biology. Which one the best? We will start the molecular biology in first years, or we must wait third year or second year? Because we know in University of Jember we have a basic course in first year, like citizenship, religion, and others. So we must wait them finish, or we must give the molecular biology course in that first years. And then, in your opinion, Prof. Anja, what methodology for best methodology for best learning methodology in condition like this, in epidemic and same condition? Thank you. Thank you for Ms. Kartika.
Kartika, I think your phone is off.
Yeah, okay, so Anja, please, you can directly answer the question. I think it is easier. Yeah.
Yeah, thank you very much for that question. So, as we heard from Prof. David, I think this has to be kind of like stairs where you start at the basic level with some basic understanding. And I think for molecular biology, there's two things that you need at a precondition. So one precondition is that you should have at least a rough idea what a cell is, and that the cell is directed by its molecular composition. So, and that leads directly to the second part of knowledge that I think is as basic for doing molecular biology, and that is the answer on what do you have to teach at the, I would say, at the first year for biology students. The second thing is that you need a minimum of organic chemistry in order to get the physicochemical properties of DNA, RNA. So you need to understand that these molecules are negatively charged, that they are rather huge compared to, let's say, glucose or something like that, that there is something like hydrogen bonds. So without that understanding, you cannot continue on the other levels. But on the other hand, it's also kind of a nice understanding, but I think that these students may already bring from school that of course you have something which is the genetic background and the phenotype that you observe in that, so that you can integrate observations at the phenotype level and bring them back to some molecular compositions. But I would say a student first semester should understand that there's a connection of the behavior, phenotype, whatever on organismic level to the molecular composition, and that this molecular composition has something to do with molecules interaction. So that was the shortest answer I could give to that very huge topic, but I think you should kind of stepwise increase that. And I strongly believe in circular learning, so that you start at one point and then it becomes more difficult and you revisit the same topic but with a deeper level of knowledge, and then you can explain a bit more, and then it is kind of a circle which gets deeper and deeper in your understanding and level of abstract things that you can have in there. And of course, that's not too easy to do that in that pandemic things, as we still know, even having all these nice, cool, super video things to do, the best thing that we learn is learning in human-human interaction. It's the same with the molecules in the cell, so they have to interact, and we have to interact as well. So that's why I would always suggest to have kind of a combination of asynchronous learning, like flipped classroom systems, where you say, okay, give material to the students, let them watch some videos, read some text, and so on, and then come together. I think one of my answers in the question and answer section goes into that direction, so that people immerse themselves into material, but you have to push them a bit. There is some pull and some push always that will really do it. So put some control levels to that. But then when you meet your students, try to do as example-wise as possible, and try to transform the knowledge into some doing. For instance, you let them read something about agarose gels, and they know, okay, I put the DNA on it and I get a picture. But when you meet them later on, give them some figures of agarose gels that are wrong, and then you discuss all together, what went wrong, how can I change the experiment, so you will see whether they understood the basic principle, and you can guide them into their real practical approaches.
So yes, Pak Ali Wafa, is there any feedback on the question, regarding the question?
Yeah, I get the point. Thank you, Prof. Anja.
Okay, then thank you for your question, and then you will be promoted again to the attendee afterward with the co-host. Okay, the next question will be from Pak Azam. Pak Azam is already in the panelist room. So Pak Azam, please, you can now ask the question directly. Pak Azam, are you still there, or? Yeah, Pak Azam. Otherwise, I will continue to the next participant. Okay. Now, while we are waiting on Pak Azam, maybe Pak Adrian Surja. Pak Adrian Surja, now you can directly. Yeah.
Thank you, moderator. Can you hear me?
Yes,
can you hear me?
Yes, yes.
Okay, I like to ask Prof. Anja.
Is it possible to turn on the video?
I don't have a good video.
Okay, okay, yeah, it's okay.
Sorry.
Okay. Yes, yeah, please continue.
I like to ask a question to Prof. Anja, because molecular biology is, you mentioned, an abstract thing, so it's difficult for the students. So what do you think that the older students help the younger students? And also, what about you said the discussions, where do you put that in your schedule? After a lecture, can they meet together in a small group and discuss it? And you mentioned also about problem-based learning. Is that a technique that you used for molecular biology? Thank you.
Yes, so directly answer the question, please.
Yeah. All right. Yeah. Thank you very much. I think it's a quite good approach to think about peer learning. So having older students teaching younger students in the practical things, and I think that it might be a good idea. And this is actually what is helpful in the real abstract thing of molecular biology, to work a lot with models and then use the things that you do in a laboratory practice in order to bring it back to what is happening in the cell. What do we imagine was the starting point for this very experiment, so that you have both kind of a practical level as well as so something that you do with your hands as well, with what is more abstract in there. And I think graphical learning and asking the students also to transfer their knowledge into some graphical schemes is a good methodological approach. And I think in non-corona times, we classically do kind of a lecture, but an interactive lecture. So it's not 1.5 hours the professor talking, and then the students may ask some questions at the end. It's more kind of having this 1.5 hour lecture with some topic, then you discuss something, then you have a task in small groups, and you come back. Today, having this online teaching formats, I think we have quite good options to do so. And this is actually what I did in my teaching during the last half a year, that I nearly fully went to the flipped classroom system, that I asked my students to really do some study before we meet. And that means I do take my slides and I put them a voice line, so that my students can listen to my lecture whenever they want to and whenever they have a stable internet connection. Mm-hmm. So considering all these technical problems that we faced during the last months. Mm-hmm. And so they are prepared and have heard my lecture, of course, without the option to ask some questions. For asking questions, I do have a forum in our internet platform, in the e-teaching module, so people can ask questions and I answer them to the whole cohort, so that everybody can really see the answers with respect to that. And then we meet from time to time in synchron formats in mostly small groups. The limitation is really if you have big groups of students to have that. And I'm pretty sure that you have to split them into small subgroups, but then you need more personnel. You need helpers that really visit those groups. But I think Big Group Button and all these tools, they have the possibilities to do that in a quite proper way, I think.
Okay. So, Pak Adrian Surja, is there any feedback? Yeah, you haven't mentioned, you haven't introduced yourself. Could you please introduce also yourself? From which faculty or from which university?
Yes, thank you, moderator, Ibu Kartika. I'm teaching physiology. So it's for pharmacy students. So for the bachelor, they come from high school and always their chemistry is not so strong. Their biology is quite okay, but for chemistry and then for organic chemistry, that is very difficult. And I think for molecular biology, you need that. So that's why I asked if the older students can help the younger, and also about the small group. And I like to get in touch with Prof. Ance about the problem-based learning, not the flipped classroom. Thank you so much.
Yes, you're welcome, Pak Adrian. Yeah, and then the next question, this is in the Q&A, but this is for Prof. David. But may I ask Bu Nur Dahlia to directly give a question to Prof. David by online? The co-host, please help me to promote Bu Nur Dahlia to allow her to to give directly her voice. Bu Nur Dahlia, are you online? Here is so okay. Yeah, Bu Nur Dahlia now is a panelist. No, not yet here. Oh, maybe yeah. She's here. Ibu bisa langsung you could directly ask your question. You are now online.
Okay.
Yeah. Is that possible to turn on your video? If it is possible, if not, you can also use only the voice.
Okay, only in voice, yeah.
Okay, introduce yourself, please. Yeah, thank you.
Okay, my name is Ulfa Nur Dahlia. I'm from IAIN Ponorogo. I'm so very interested with a presentation from Professor David, how the can motivation student to collaboration research so we can have the good result, and so and how we can avoid the underestimate about the competency from our student. Okay, like that. I think is enough. Thank you.
Yes, okay. Pak Prof. Davi, please, you could directly answer the question.
Yeah, thank you, Ibu. So actually, when we talk about interaction and student activities, of course, we need to deal with the teaching methodology or teaching models in the classroom. That's the first one. We have a lot of teaching model. For example, in this case, we have, this one I can share. Yeah, small group discussion, role playing and simulation, for example, discovery and inquiry learning, contextual teaching and learning. And we insert one type of teaching model, namely research-based learning. So that I said before, research-based learning, what is the difference of research-based learning and project-based learning or problem discovery, inquiry learning? In research-based learning, there exists the involvement of research group members into a classroom. So when we have a research group in in the university level or in the program study level, of course you have a member. How many members of research group do you have? So you should ask them to come and to join in the classroom. Not all the time, of course. Sometimes you should schedule them. The first week, you should ask one person to come or 2 person to join the second week. And finally, they can motivate the student who are joining the classroom to do a real research. But if you alone doing teaching or delivering material in the classroom, it is very hard to motivate them to do a research, of course. So by involvement or involving of the research group members, of course, you can give a strong motivation doing the real thing or doing the research activity in the classroom. That is the first one. Why it is possible? Because in the research group, normally there is a permanent research member and there is like postgraduate research group members, which is what might be the senior of their student can be also asked to join the classroom. And there can be a pair learning, a pair student, or give them a motivation, because they give them a testimony how very nice to do coursework as well as doing researching. So this one, that's a strategy. By using research-based learning, you are not alone in the classroom. Even you have a team teaching, for example, at most team teaching only 2 or 3 members. But if you have a research-based learning implementation in the classroom, you can ask a research member as a guest lecturer in the classroom. That's the student can have a high motivation, because they can be a best practice. They can give a best practice in research. They can be a model in research, because their age normally quite similar with them. But with the lecturer, especially the senior lecturer, they have a very different age, maybe go into further and their activity and their comfort zone. But if they do a peer research, ask them as a peer researcher, I believe it will be easy to motivate them to your research. Of course, I don't want to. I'm not talking about the basic elementary level of subject courses. At least you can do a research-based learning in the subject courses at least at the 5th semester, 6th semester, 7th semester, and the last one 8th semester. I believe all the subjects courses already associating with the research activity or research of interest. But for the elementary coursework, I believe you need to have like a normal basic fundamental knowledge should be introduced to have a better understanding of student cognitive. Thank you very much. I think that's enough.
Oh, yeah. Thank you so much, Prof. David, a very elaborative answer. And Bu Nur Dahlia, is there any feedback you wanted to say?
No, I think it's enough. It's so very, I get the point how to make our student can collaboration. Okay, thank you.
Okay, thank you so much. We are still having so much time actually. So I invite, if there are still questions from the attendee, from the Q&A, Anja has been answering all the questions. But if not, then you could also raise your hand here. Okay, while waiting for your information, there are many commentary in the chat room about the real practice in online teaching. And I'm very happy to inform you there will be for tomorrow. So we are really eager about the tomorrow topics, how we do the online teaching, not only the online teaching in terms of theoretical things, but also in terms of how we do the lab things. That is now really we have to really do less interacting with the students. How do we do this kind of teaching? So tomorrow, yeah. So I saw here there is no further question, and the Q&A has been also all answered. Okay. Maybe so we are going to go to the end of the webinar. So for your information, tomorrow the webinar will start at 1 o'clock Indonesian time, which is 8 o'clock in Germany, right? Yeah. Yeah, so and we will have tomorrow Holger Rehman and Marlies Witte as speakers. And there are many questions about the presentation and the slide. As I already wrote so many times also in the answer, that we will send you all the A module by the end of the webinar, and you don't have to worry about it. So until now we have about 200 now it's less than 200 participants in online in Zoom, but we have about 100, now it's more than 100 viewers in the YouTube. So please, you could also comment in the YouTube so that we could also share all the material from today and as well as from tomorrow from our webinar. Don't worry about that. Just give the comments and then we will send you by email. So I think now we come to the end of the webinar, and thank you so much for your attention, and thank you so much for all the speakers. It's really our honor here to have you in our webinar. Thank you, Prof. Angela Bes. Thank you, Prof. Davik, for sharing with us many experience. And we really hope to see you again in the next webinar. And now I come back to the end and wanted to give back to the Master of Ceremony, Bu Enda. From me, wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Renat Kartika Senjarini, for to moderate this discussion for today. And I would like to announce to the all participants, I would like to remind you all that this is a 2 days webinar, so please stay because the feedback form that you ask will be shown at any time. So we hope that you have the whole comprehensive material. So we will still have tomorrow session. So until we meet again tomorrow, and to all the speakers and the panelists, I would like to say thank you for today. I'm looking forward to meet you again tomorrow. Thank you. Good afternoon and wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Waalaikumsalam. Thank you. Thank you.
Ya, makasih Prof. David. Thank you, Holger.
Thank you.
Malik.
Bye. See you tomorrow.
Bye. See you tomorrow.
Yeah.
Yeah. Yeah, you may leave the webinar room if you wanted. Okay. Bisa leave. Okay. Ya. Ini Pak Rektor masih on. Nggih, paling nanti saya tak kontak dengan Pak Sekretaris Mbak Sari nanti terkait dengan meeting besok. Nggih, sampun nggih Bu Enda. Jadi bahasa Jawa ini sudah balik soalnya. Nggih, terima kasih semua partisipan. Bapak Ibu sampai ketemu besok nggih. List attendees akan diberikan. Ini ada lagi yang bertanya list attendees Bu Enda. Besok ya.
Tidak ada list attendees ya Bu. We don't have the list attendees, so we will have the form for the feedback, and it will be displayed at any time. So please stay. This is a 2 days webinar, and you will have your certificate directly to your email once you fill the form. Thank you.
Ya. Jadi Bapak Ibu begitu ngisi feedback, kelihatannya mungkin besok mau diberikan, langsung akan dapat sertifikatnya lewat email Bapak Ibu, ya langsung gitu nanti. Ya. Nanti kalau e-modul kita akan berikan kepada semua yang sudah register dan sudah terrekod di hari ini dan besok. Ya. Sudah nggih Bu Enda, LP2M itu Mas Andika, terima kasih nggih. Saya pamit dulu. Terima kasih Bu Enda, Bu Nur.
Thank you all. Thank you all the participants. Good afternoon.
Assalamualaikum.
Waalaikumsalam.
Warahmatullahi wabarakatuh.
To host, could you please end this meeting, please? Thank you.
It's only. Ini saya